I would like to begin by acknowledging that the Wurundjeri people of the Kulin Nation are the traditional owners of the lands at Parkville, Kew, Bundoora and the Elizabeth Street campus on which WeHi operates. We pay our respects to the elders past and present and embrace their continued connection to the places where WeHi staff and students work. We recognise that Aboriginal and Torres Strait Islander peoples as the first scientists on this continent who ask questions and especially look to the land, the sky, the animals and plants for answers. We acknowledge the ingenuity, resilience, design capability, collaborative and creative thinking of the First Nations people that enabled them to thrive and ensure the voices and designs of the first scientists were passed from generation to generation. We respect that as the oldest living generations of teachers, artists, engineers and scientists, how they understood and continue to understand the world has benefited and will always benefit all of us. So today, it's my very great pleasure to introduce Brigitte Duckworth, who will be presenting her final PhD seminar. Brigitte will be familiar to many of you. She started WeHi as an undergraduate student, working with Gabrielle Bells and Lisa Milkey. She then performed her honours research with Gabrielle, Phil Hodgkin, Sue Heinzel before joining my lab as a research assistant. During her time at WeHi, Brigitte has been actively involved in outreach activities and served on several committees, including WESA and the Centre for Dynamic Imaging Special Interest Group. For those of you that were here during the pandemic, you will probably remember Brigitte's dedication to making face masks, which could be seen all over WeHi, and I think really increased our sense of community at that time. It was really much needed. This work also continued Brigitte's dedication to supporting and raising funds for CJD research, which is a cause close to her heart. So the plan was always that Brigitte would work as a research assistant for a few years before returning to Gab's lab to do a PhD. So you can all probably imagine how thrilled I was when I advertised a student project on the generation and maintenance of T-cell memory. And Brigitte said that she was keen to take that up. During her projects, as you can already see, in close collaboration with Verena Wimmer and the Dynamic Imaging Facility, Brigitte mastered tissue clearing and light chain microscopy. Her imaging work has been featured in Zeiss brochures, Art in Science, and the cover of Nature Immunology, where the first story from her PhD has been published. Brigitte is also a very skilled science communicator. She's won prizes for presentations, including the Vin Young Investigator Symposium and 3-minute thesis competitions at IGV, WeHi and the University of Melbourne. She's also been awarded the Sydney and Joan Pesca Graduate Prize for the International Cytokine and Interferon Society and the Pfizer New Investigator Award at the ASI National Meeting last year. And for those of you who don't know, this is really the most prestigious award that an early career researcher, immunologist can receive within Australia. So believe it or not, with that introduction, I could actually go on for a fair bit longer. But I would like to finish by really saying to Brigitte, thank you for joining the lab. Thank you for staying in the lab to do your PhD. I think we've made a really incredible team and it's really been fabulous to watch you mature as a scientist. I'm very excited for everything you've achieved and everything that's to come. And to the rest of you, I'd like to say, sit back, enjoy. This is going to be a good one.
After that, thank you all for coming. So today, as Jo mentioned, I will be taking you through a body of work I performed during my PhD, really looking at the spatial requirements for T-cell memory. And when I talk about T-cell memory today, I'm going to be talking about CD8 or cytotoxic T-cells. These are an incredibly important arm of the antigen-specific adaptive immune response, which are key in cancer and tumour surveillance, fighting intracellular bacteria, and clearance of viral infections. When they misbehave, these cells contribute to autoimmune disease, and currently they present one of the largest barriers to strategic vaccine design, with most of the vaccines we use today targeting B cells and antibodies with a really poor responses in viruses which require CD8 T cell responses from vaccines. And with this in mind, we believe that understanding the fundamental drivers of T cell memory will be able to strategically use this to design better vaccines and therapies in the future. So immune memory. Here we have a naive CD8 T cell. When it's activated, it encounters its cognate antigen through infection or we vaccinate. This T cell is turned on and it multiplies to reform a responding population, which is made up of effector cells, which go on to clear infection and then die, and memory cells, which stay around long term. So once infection is cleared, we're left behind with this long-lived pool of memory cells, which consist of us for up to our whole lives in some cases. So these memory cells protect us upon secondary challenge where we encounter the same pathogen again. These cells are activated and they respond more quickly and more efficiently to infection, clearing infection before we even show symptoms in most cases. And it's really this basis of forming long-lived immune memory with which we manipulate for vaccines. And so in my PhD, I've looked at two areas of this immune response curve. One is really early in infection. So when we have that initial activation of our T cells, how do they make those fate decisions to become effectors or memory precursors? And really, how does this balance between these two fates influence the outcome of infection? And secondly, looking in the steady state, we have these long-lived memory cells and they live with us for a really long time, and we still don't really know fully how these cells remain so long-lived, the homeostatic proliferation that goes into that, and how we can maintain them longer. And so I'm asking these two questions: how do we make memory and how do we maintain it in the context of the lymph node? So the lymph node is a highly organised immune structure, and it's organised into these compartments which separate immune cells out. So we have our paracortex, we have our B cell follicles, interfollicular regions, cortical ridge, and our medulla. And it's really a balance of cell migration and movements between these spaces and in and out of the lymph node that regulates our immune response and facilitates appropriate immune cell interactions to drive that responding immune population. And so this migration is largely mediated by chemical messengers known as chemokines. And so chemokines are produced in one region of the lymph node and detected on by chemokine receptors on immune cells, such as T cells here. And when this chemokine receptor detects chemokine, those cells migrate towards the chemokine along a chemokine gradient. And so this really facilitates controlled movement of cells around the lymph node, really orchestrating this cell migration in response to infection. Take a second to load. Yeah, beautiful. So for my first question, we really asked what are the spatial requirements for early T cell memory precursors? So how do we make memory? So here we have a lymph node, and we know a lot about where different T cells are located in the lymph node. We know that we find naive T cells in the paracortex, and studies have shown out in these interfollicular regions we find effector cells, and this makes sense as this is a region of high inflammation. It's where we have antigen drainage and a lot of antigen presentation during infection. However, there are questions remained about whether or not cells are required to go to this space in order to become effectors. And so we had a hypothesis that perhaps there were distinct lymph node niches where cells needed to go in order to imprint these fates. And so looking first at these effector cells in the periphery, we asked, is T cell differentiation dependent on this peripheral migration? Now, firstly, to look at effectors, we're using TBET as a marker of effector differentiation. TBET's a master regulator, which is important for effectors. If you don't have it, you have reduced effectors in response to infection. And so we use reporter mice which report the expression of TBET, known as ZS green TBET mice. And so these mice they have endogenously red cells, and when these cells express TBET, they turn green, so they become red and green. Now, we cross these cells to P14's transgenic mice. And so in these mice, they have a transgenic T cell receptor, which specifically recognises the infection LCMV Armstrong. So it recognises the GP33 peptide from this virus. So when we transfer in these red antigen-specific naive T cells into black 6 mice, we infected with that LCMV infection, and then we looked across the time course of the infection from day 0 to 8 to see when these effectors emerge. And so here we've got some FACS plots. We've got ZS green versus KLRG1. So KLRG1 is a marker of effectors, so we use this as a marker of when effectors start to differentiate. So as we can see at day 4, we have TBET being expressed by these cells, followed by KLRG1. And by day 6 and day 8, we really see this nice effector population forming. And so what we're showing is we've got red cells, they express TBET, and then they're expressing KLRG1. So we're using these as our effectors. And so when we look at the location of these cells in the lymph node at day 6, here I'm showing an inguinal lymph node that's been cleared, and we've got our reporters for all of the transferred cells in pink and our TBET expression in cyan. What we can see is this cyan expression is really limited to the periphery of the lymph node in these almost interfollicular pockets, and we can see this around the lymph node. So rather than just taking my word for it, we quantified these images. So I established this quantification pipeline in the lab with collaboration with Thomas Boudoir, who was a visiting scientist in the Rogers lab. And we set up a really simple mechanism to calculate the density of cells in 3D space. And so we defined any cell that we found in the middle of the lymph node with a value of 1, and any cell in the outside of the lymph node with a value of 0 in the periphery. And we can map this quantity across the lymph node in 3 dimensions, and this normalizes for changes in size between lymph nodes. And what we found was that the highest density of our transferred cells were in the centre of the lymph node, whereas TBET expression was shifted towards the periphery. So we see that these cells start expressing TBET closer towards the periphery of the node. We performed statistical analysis on this, and so the way that we read these graphs is that this is the p-value, as indicated on the right, and then we assigned a significance of 0.05. So essentially this is just multiple t-tests between these points, and you can see anything above this dotted line is significant. And so I'll be using these plots throughout the rest of my talk. We can also look a little bit closer at specific regions. So for example, looking in the periphery at the interfollicular regions, and we can see that there is a significant increase in our TBET expression compared to when we look in the centre of the lymph node where we see a decrease in TBET. So overall, this led us to propose that based on this TBET expression, effector T cells differentiate in the lymph node periphery. So our next question was, how do they get there and how is this regulated? So one of the targets of TBET in T cells is the chemokine receptor CXCR3. So we looked at how CXCR3 was regulated in comparison to TBET across the time course, and we saw that as TBET's upregulated, CXCR3 shows a slightly delayed but very similar kinetic. So what we think is happening is the cells are putting on TBET, they're expressing CXCR3, and then this is followed by that effector differentiation at day 6. So what is CXCR3? It's a lot of chemokines, so I'm just going to take you through a couple today. So CXCR3 is one of the most studied chemokines in T cell biology. It binds 3 ligands, CXCR9, 10 and 11. Black 6 mice, which we're using throughout this study, do not express CXCR11 due to a frameshift mutation. And there's been work in our lab which has shown, done by Len, which has shown that CXCR11's role in this context is largely redundant. So we'll be focusing on CXCR9 and 10 today. So CXCR3 has a range of roles in T cell activation, survival adhesion, cell proliferation, and specifically it plays a key role in migration and effector function and trafficking to inflamed tissues. So far I've shown you we have naive T cells in the paracortex. They are activated, they upregulate TBET, followed by CXCR3, and then they migrate out to the periphery where they become effectors. So we think this might have something to do with their expression of CXCR3. And so really to address this, we asked what happens if these cells lose CXCR3? What happens to this migration and what happens to their fate? So to answer this question, we generated mice deficient in CXCR3. We crossed them to GFP reporters so we can see the cells via microscopy, so they'll be green. And we also crossed them to our transgenic P14 model, so they're antigen specific. We then transferred these cells into black 6 mice, infected with LCMV, and looked at day 6 where we really see that emergence of effectors, and looked at these cells via flow and also microscopy. And so when we asked the question of where do these cells go, we found there was quite a difference between our wild types and our knockouts. As you can see in these images, our GFP cells in yellow have a reduced migration out to the periphery in the knockouts when we compare these to the wild types. And we can see that they're restricted within the T cell paracortex beneath the B cell follicles in blue. So it really looks like this loss of CXCR3 is driving a retention in the T cell paracortex. So we quantified these images. So for a lot of my talk, I'm going to be showing you cross-sections of these nodes. So these are intact, like a full orange, and then a cross-section, which is basically just a section through the middle. And what we can see really nicely is this beautiful peripheral migration in the wild types and this retention in the centre and the knockouts. And when we quantified this, this was the case. So we had this significant decrease of cells in the periphery and an increase in the centre in our knockout mice. And we can see this here in the reduction in interfollicular regions and an increase in the paracortex. So given that CXCR3 was stopping this peripheral migration, we wanted to see how this affected effector fate. And so via flow cytometry, I looked at various markers for effector cells and also memory precursors at this early time point. And what I found was that when we knock out CXCR3, we have a massive reduction in the number of effectors we find in these lymph nodes. Conversely, we see an increase in the expression of CD62L, which is a marker of memory. When we look transcriptionally at these cells, by sending for some transcription factors, we found that these cells were upregulating TCF1 in the knockouts, and also that this TCF1 was co-expressed with CD62L. Looking at cytokine profiles, and we found that the molecular granzyme B was decreased on these knockouts, which is associated with effector differentiation, and also IL-2 was increased on the knockouts, which we would see in memory cells. So this combined with a whole other panel of markers, which I won't go through today, we really define these cells as a stem-like memory precursor at this early time point following infection. And so it looks like in the absence of CXCR3, we're seeing an increase of this really potent stem-like pool of memory cells, which really promote self-renewal. They suppress terminal differentiation. So when reactivated, they're able to give rise to a large range of responding cells. They play a key role in the control of chronic infection, and they're also analogous to cells which control tumour growth following checkpoint blockade inhibition. And so we were really interested in how we can skew cells towards this fate. And so far we've looked at these cells in the periphery. When we lose CXCR3, they're reduced. They instead stay in the centre and they become stem-like memory cells. And so we're really curious in the lymph node how chemokines may be actually regulating this migration between these two areas. And so we looked really firstly at what was really driving this peripheral positioning. And so, as I mentioned earlier, CXCR3, the chemokine receptor upregulated on our cells, binds two ligands, CXCR9 and CXCL10. And so during her postdoc, Jo generated mice which report the expression of these ligands known as REX3 mice, where we have CXCR9 being reported with RFP and CXCR10 being reported with BFP. And so we can basically use these mice to see where the cellular sources of those chemokines are in our lymph nodes. So we infected these mice with LCMV and we looked at day 6 following infection to see where these cellular sources were. And so this is an intact lymph node at day 6. We can see CXCR9 and 10 in pink and cyan, and then our blood vessels in yellow and our B cell follicles in blue. And just to look a little bit closer at these cells producing these chemokines, what we can see really nicely is that CXCR9 is being produced throughout the lymph node, so with a peak in the lymph node centre, whereas CXCR10 is really being produced in the lymph node periphery in these interfollicular regions. And you can see this here with this quantification, with this really big peak of CXCR10 producing cells in the lymph node periphery. And so we asked the question, what if we were to lose these chemokines? Would this affect cell migration, and how would this affect fate? As these chemokines are produced in 2 different regions of the lymph node. So we generated knockouts for CXCR9 and CXCR10. We transferred in our transgenic T cells. We infected with LCMV, and then we looked again at day 6 to see how this affected cell fate. And what we found was that in the CXCR9 knockouts, there was no change to the number of stem-like memory cells we were able to recover. However, when you knock out CXCR10, it really looks like our activated T cells are being skewed towards a stem-like memory fate. When we compare this with the location of these cells, this really makes sense. So what we found was that in the CXCR9 knockouts, there was no significant difference in the way where these cells migrated following infection. However, when we knocked out CXCL10, we saw a shift of these cells back to the lymph node centre, very similar to what happened when we knocked out CXCR3. And so together this let us propose that it's really CXCL10 that's driving this effector positioning to the periphery. And so this is where our model this is where we led to our model. So we've got CXCR10 being produced in the lymph node periphery, and this is driving effector differentiation. But our next question really was, what is driving positioning of cells in the lymph node centre? If it's not CXCR9, is there another chemokine that's really retaining these cells? And so given the role we found of TBET, of upregulating CXCR3 and driving effector differentiation, we wondered whether or not TCF1 in our stem-like memory cells were performing a similar function. So we turned to look at the downstream targets of TCF1 to see if there's any chemokine receptors there. And we had some ChIP-seq data from our collaborator Lisa Milkey over at ONJ, where she looked at the binding sites of TCF1 and found that it binds the promoter of CCR7. So CCR7 is another chemokine receptor. I told you there's a lot of them. And CCR7, when we looked at expression on our stem-like and effectors, we found that it was increased on our stem-like memory cells in both our knockouts and the wild types. So we proposed that it was perhaps CCR7 that was driving cells towards the lymph node centre. And this was really nice, as we know the lymph node centre is really abundant in the ligands for CCR7, CCR21 and CCR19. So to see whether these cells migrated preferentially towards CCR21 when compared to effectors, Raymond performed some transwell chemotaxis assays where we load the chemokine into the bottom well, we pop our cells into the top well, and we can measure the migration of these cells towards the chemokine, and this gives us a migration index. And what Raymond showed in this experiment was that our stem-like memory cells had a higher migration index towards CCR21 when compared to our effectors. So together this allowed us to propose this model. We really demonstrated that CXCR3 is driving effector differentiation, migration to the periphery and effector differentiation here, whereas we propose that it's likely CCR21 and CCR7 that's leading to the retention of stem-like memory cells. Now, this is just a brief overview of work we've published, and further we really started to investigate how we can skew cells towards these two fates. And we plan to incorporate this into vaccine design in the lab using mRNA vaccines. We're really trying to see early in infection if we can skew cell fates and change the outcome of the immune response. So that was how we make memory. Our next question really was, how do we maintain it? So we're shifting gear here now, and we were previously looking at memory precursors, and now I'm really interested in looking at long-lived, established CD8 memory T cell populations. So CD8 T cell memory cells come in a range of flavours, and they're really distinguished by where we find them and their circulation properties. So within our secondary lymphoid organs, such as our lymph nodes and our spleens, we find central memory and a subset of central memory, stem-like memory cells. And these are largely confined to circulating in these secondary lymphoid organs. Out in the periphery, we find memory T cells. So these are circulating, ready to encounter antigen. And we also find tissue-resident memory cells, which have more recently been described over the past few years. And these take up residence, as their name suggests, at barrier sites to infection and disease, such as the skin and the mucosa. And so together, this really forms up the large memory pool we find in the body. So I'm specifically interested today in looking at the central memory T cells, which make up the bulk of the cells in the lymph node. So as I said earlier, we know quite a bit about where some T cells are found in the lymph node. So naive T cells are found in the paracortex. But what's largely debated in the field is where exactly are central memory cells located. So for a long time, it was assumed, as they are quite similar to naive T cells and a lot of their properties, that memory cells were found within the T cell paracortex. However, some more recent studies over the past decade have really thrown this theory into chaos. And so there's really kind of a controversial question in the field of where exactly these cells are located. So in this one study performed in 2013, authors transferred in antigen-specific cells into black 6 mice. They infected with a vaccinia virus, and then they looked at day 60, so long after memory's been established. And just before harvest, they transferred in naive T cells to see if there was a difference in where these cells were located. And what they found was in the draining lymph nodes for this infection, that while naive T cells were found in the paracortex, the memory cells were actually positioned more peripherally. So there was a higher density of cells out in the cortical ridge beneath these B cell follicles. And when they quantified this, they showed that there really was this distinct shift of memory cells towards the lymph node periphery. And so they proposed, so they tested whether or not they could shift these cells. They knocked out various chemokine receptors listed here. They tried blocking CD44, and they did chemical inhibition of EBI2, which is really found abundantly in this cortical ridge. And none of these interventions disrupted this location. And so they proposed that there was perhaps an advantage to these memory cells being pre-positioned closer to where there's antigen drainage, closer to effector cell entry. However, they didn't really explore this further. At the same time, another paper came out where they generated memory in a different context. So rather than generating them in vivo in mice, they did an in vitro culture where they skewed cells towards a memory fate, and they co-transferred these with naive P14 cells. And what they found was that in this model, the cells did not go to the cortical ridge. They co-localized with the naive cells. And so they proposed that naive and memory really occupy the same region of the lymph node. And this is really where the field was left. And it's this work that we kind of based our hypothesis on to really answer this question of where are central memory T cells located. So we proposed that central memory T cells occupy a distinct region of the lymph node, and that this region maintains memory longevity and function and gives them an advantage upon secondary challenge against naive T cells. And so to really answer this hypothesis, I had three major aims for this project. The first was to really characterise this location and really resolve this controversy in the field of where exactly are these cells located. Secondly was to identify the cellular interactions between our memory cells and their environment, which maintain their longevity and maintenance long term. And lastly was to determine if these cells even need a location. Is this important for their secondary recall? And really, if it is, how can we use this for therapeutic benefit? So how can we target cells towards this region following vaccination to really boost and enhance immune memory? So for my first aim, my first question was, well, let's generate memory in vivo. And so to do this, I used our naive antigen-specific GFP-labeled P14 cells I used in the first part of my talk. We transferred these into black 6 mice. We infected with LCMV Armstrong. So this is an acute infection, which is cleared. And then we looked at day 60 at harvest. And just phenotyping these cells via flow cytometry, I showed that at day 60, we have around 90% of the memory cells found in these lymph nodes are central memory, with a small proportion being those circulating effectors. When I phenotyped these cells, they were indeed memory cells. They expressed all the markers we'd expect, IL-7 receptor, IL-2 receptor. And what was really important was this is a really homogeneous population. So with over 90% of these cells being central memory, when we're looking at our imaging of these lymph nodes, we're really looking at central memory cells. So we turn to location next. So this is an inguinal lymph node at day 60 following infection. We've got our labeled GFP cells in yellow, and I've stained for the paracortex, so stained for the CD8 T cells in pink, and the B cell follicles in blue, really to mark out those major structures within the lymph node and define the cortical ridge. So I've quantified these two structures, so the B cell follicles and the T cell zone, to really give us an idea of the structure of this lymph node. And when we overlay the density of our memory cells, we can see that they're really peaking in highest density at the interface between the T cell zone and the B cell follicles, i.e. the cortical ridge. And we also see cells out in the lymph node periphery. And so from this, I concluded that central memory T cells are found at the highest density in the cortical ridge at day 60 post this LCMV infection. So my next question was, are these distinct from naive cells? So similar to that earlier experiment I showed, I transferred in cells. Instead of infecting, we just harvested 2 days later, and we had it look where these cells home to. And what we found in these naive lymph nodes, that the naive cells were indeed found in the T cell paracortex. Comparably, when we looked at memory, we found these in the cortical ridge. And when we quantified these and compared them, we can see that there really is a significantly distinct location for our naive cells in purple and our memory cells in green. So as that first paper proposed, central memory location is distinct from naive. And so lastly, I've been looking at LCMV for this infection, which is systemic. So this infects the whole mouse. So this gives us a lot of tissues to look at. And so I looked at all of the lymph nodes from these mice, so inguinal, brachial, axillary, popliteal, iliac and cervical, and I found across all of these lymph nodes, this peripheral positioning at the cortical ridge was conserved. So it is not just specific to the inguinal lymph nodes I've shown so far. So having characterised this memory, my next questions really were, what other factors might be driving T cells to this location. And so the controversy in the field from those two papers suggested a few reasons. Maybe it's residual antigen from LCMV infection, maybe it's infection specific. And so we really wanted to tease apart these questions. So firstly, I wanted to see whether there was a difference in the way transferred cells trafficked in mice which had previously been infected. So LCMV infects the lymph node. It's a highly inflammatory virus. So I wanted to see if naive T cells went to the same locations just as a control between these two conditions. And so what I found was that in both the naive hosts and the mice which had been infected and recovered from LCMV, there was no difference into where these cells trafficked. So secondly, I wanted to address this question of those in vitro cultured cells and where they went. So to do this, I worked with Raymond in our lab, who generated some skewing assays to skew our transgenic T cells towards memory fate. And so we transferred these central memory cells into naive mice and then also previously infected mice. We again saw there was no difference into where these cells trafficked, and they both went to the T cell paracortex. So these are quite simple but quite important experiments for this work, because one, it showed this trafficking isn't influenced just by having infected lymph nodes. So having an infected lymph node wasn't enough for these cells to want to go to the cortical ridge. And secondly, even if we were to skew them towards memory in vitro, they still don't want to go there. So it's not just a matter of pairing a recovered lymph node with a memory cell. There's something that happens intrinsically in the T cells when they're primed in the lymph node in an infection that drives them to the cortical ridge. So one of the other questions I had was whether or not this location was influenced by cell trafficking. As I said earlier, we have cells circulating throughout the body, and whether or not blocking lymph node entry and exit would disrupt where these T cells were located. And so to do this, we treated the mice with CD62L, which blocks lymph node entry, and FTY720, which blocks lymph node exit. Slide takes a bit to load. Yeah. And so we set up memory, as I've shown previously, and then just prior to harvest, we did daily IPs of these blocking treatments. And what I found by imaging, that there was no difference in the location of these cells. So despite blocking T cell entry and exit from the lymph node, this location remained stable. And so it doesn't seem to be that these cells are going out to the edge to leave. It seems to be a relatively stable location in both the control and the blocked groups. So having characterised memory so far in the context of LCMV, my next question was, is this conserved in other infection models? So to address this question, I sourced a range of GFP transgenic mice for various infections, and then had a look at influenza. So this is a mediastinal lymph node, which is a draining lymph node for influenza. But I must say, for all these infections, I looked at all the lymph nodes in the body and found the same location. And so looking at the OT1 transgenic cells in these mice, we see this peripheral positioning towards the edge of the lymph node. Following herpes simplex virus, which is a skin draining infection, looking at the brachial lymph node, again, we saw this peripheral positioning and this peak at the cortical ridge. And lastly, looking at bacterial infection, listeria, which is systemic, again, we saw this really nice clustering of our T cells at the cortical ridge. And so combined, this data was really interesting to me, and I was quite excited when I got these results, because it shows that this location is really conserved across these different infections. Despite they drain differently to the lymph node, they have different dissemination, antigen presentation, these lymph nodes all had this conserved location, which really strengthened this idea that these cells may need this for their long-lived memory and longevity. So far I've characterised this location. And my next question was really to interrogate what's happening here at the cortical ridge that's making these cells want to stay there. And so we hypothesised that this location may be providing something that really supports the cells. And so we really wanted to look at the cellular partners and interactions between our memory cells and this microenvironment. So what cells are these co-localising with? So to answer this question, I used confocal microscopy to really examine a range of different dendritic cells, macrophages, stromal cells, to try and tease apart this environment visually. And so one of the markers we looked at was CD301B, which is a marker of a subset of CDC2s, which have been really well described in the skin and other barrier sites is supporting those tissue-resident memory cells I talked about earlier. So they provide survival signals and really support that tissue-resident niche. And we hypothesised whether or not they might be providing a similar environment to our T cells in the lymph node where they're supporting that niche. And what we saw really nicely was that this is just a cleared lymph node, and I've just stained bulk T cells to show the paracortex, but these CD301B DCs really do line the cortical ridge where we see our T cells. So we looked a little bit closer at this region. So this is just zoomed in on the cortical ridge, and you can see the B cell follicles outlined here. And what we saw was that there was this really nice overlay of our memory cells and these DCs. And when we looked a little bit closer, beautiful, we could see that these T cells were in contact with these DCs in this region. And so we proposed that perhaps there was an interaction occurring between these cells that was driving our T cells to this area. And so I set up a quantification pipeline with Lockie to really look deeper at these multiplex lymph nodes to quantify different cells in this region and their distance from the memory cells. And so this is just some preliminary data. But one of the first questions we asked was there a difference in the distance of these DCs to our memory cells compared to naive? And what we found when we measured this, that looking at the minimum distance from DCs, that the central memory cells were indeed closer to these DCs than our naive T cells were. Secondly, I also found there was another subset of CD207 Langerhans cells that were found in this region. And so we wanted to see if there was a difference between the distance of our T cells from these two DC subsets. And what I found was that the T cells were indeed closer to the CD301B DCs compared to CD207. And so we're going forward and applying this quantification to a range of different markers to see if we can start to kind of get an idea of what cells are close to our T cells in this region. And so this is one of the ways that we're kind of attacking this question. And the other is, at the moment with these DCs as a target, I have these CD301B DTR mice, so we can generate memory and then we can treat the mice with diphtheria toxin and specifically deplete the dendritic cells. And the question there is, do the T cells move? And that'll give us an idea if these DCs are important for that location. And secondly, we are performing single cell RNA sequencing of our central memory cells and then also a bulk dendritic cell population. And we're collaborating with Melissa Davies and Xin Wei to really look at predicting novel receptor pair interactions that may be occurring between these two cell types within the lymph node. So another cell type which makes up a bulk of the lymph node are stromal cells, and these really comprise the microarchitecture of the lymph node and play an important role in not just the trafficking of cells, but also comprise of supportive niches. And so here I've got another zoomed in section of the cortical ridge. So I've got our B cell follicles here, just so you can orientate yourselves. And I've stained for a range of stromal markers. And what I found in this region, if we zoom in a little bit closer, our T cells really seem to be quite closely associated with these fibroblastic reticular cells, which are co-stained for both podoplanin in blue and ERTR7 in orange. And so I was curious about the role these stromal cells play with T cells, as they interact with T cells universally throughout the lymph node. But this really interesting paper came out in 2020, which showed that if you specifically deplete IL-7 from these stromal cells, you see a loss of memory cells in the lymph node, but not a loss of naive. And so this was quite interesting, as it led me to kind of think maybe these cells are producing IL-7, which is producing memory cells in this niche. But we have a lot of further research to do on this question. And so where this work is at the moment is really in the quite exciting part. So we've got this ongoing work where we are further dissecting the cellular partners within this niche using photoconvertible KD mice. So if you saw our collab pitch with Verena and I, Verena's making real headway using these mice. So what we do is we use a laser to colour in different regions of the lymph nodes. We can then sort these photoconverted cells and perform single cell RNA sequencing to look further at these real these kind of niche specific areas and look at what cells are present there, who's interacting. And one of the advantages of this is we can do it on quite thick sections, so we can do it in 3D. And secondly, we're currently on a wait list to do spatial transcriptomics with nanostring on this work, to use to really look at identifying novel cellular partners within this memory niche. And so really future questions for this project, importantly, are how do changes, so once we've identified potential regulators of this location and whether we can disrupt these, whether that's through CRISPR gene editing or through monoclonal antibodies, if these cells move, how does this affect the secondary immune response if we were to re-challenge these mice? And secondly, can we manipulate this positioning, really importantly, for strategic vaccine design? So how can we manipulate this peripheral positioning for designing better vaccines which elicit stronger immune memory? So today I've taken you through two very different parts of the immune response curve, but both very important. One is how we make memory. So I've really demonstrated that memory is really dependent on the spatial imprinting within the lymph nodes. So we see effectors differentiating out in the periphery, whereas memory cells really like to be in the T-cell paracortex. And we're exploring further how we can increase retention in the paracortex and drive stem-like memory cells in response to vaccines. And secondly, I've started to explore how memory is maintained. So I've identified a novel niche that really, and resolved this outstanding question in the field of where memory cells are located. And I've started to identify potential cellular partners which may be regulating this, and have worked in the process to further tease apart this microenvironment and look at ultimately how we can improve better memory for therapeutic benefit and use this for strategic vaccine design in the future. So that was a lot of work, and it absolutely was not done just by me. And there is a huge number of people to thank, and obviously with time today I can't go through everyone, but I really wanted to highlight some key players who played a really key role in my PhD over the past 4 years and my past 9.5 years at WeHi. So I'm going to leave you to last, Joe, if that's all right. So I just want to start with Gabrielle, who's been my co-supervisor throughout this project, and also my supervisor throughout honours. It's been really delightful to grow as a scientist with you under your leadership. I'm really, really grateful for your mentorship and guidance, and this has continued even though you've moved to Queensland, and I really hope that it continues onward throughout my career. I'd like to say a huge thank you to the Groom Lab. It's an incredibly special team to be a part of. Everyone really works together, and I know I can ask anybody to help me with something, and it's a really fun and brilliant and collaborative team. And I really want to highlight Carolina. So any mouse work you see here has Carolina's hands on it in some way. I'm really grateful for your technical expertise and the fact that you'll come in on weekends to help me with injections, and none of this work really would be possible without you. A huge thank you to my PhD committee, Phil, Laura, Charlene and Mark. It's been an absolute privilege to discuss science with you after every seminar. I really appreciated your mentorship and guidance, keeping me on track when I get distracted by interesting findings, and really helping me bring this PhD home. And these are relationships I really hope to continue onwards after my PhD is done. Huge thank you to everyone in the Immunology Division, people who've supported me throughout this time, a whole range of people who's provided reagents. I can't go through everybody. A huge thank you to the Rogers Lab and the Centre for Dynamic Imaging. And I really want to shout out to Verena. You took me from not being able to turn on a microscope to independently running all of my own experiments, and your skill and expertise is really evidenced in the paper that we got to publish together. Huge thank you to Bioservices. We have a lot of mice in this experiments, and so they've been incredibly important in maintaining and keeping them safe and keeping these experiments ongoing. And to all our collaborators, both at WeHi and externally. Lastly, I want to thank Joe. So Joe has been an incredible mentor and supervisor to me, both as an RA and through my time as a PhD, and I'm really grateful that that relationship has turned into quite good friends as well. I come to you for a lot of questions, science and non-science, and it's a relationship I really am looking forward to continuing both through our scientific careers. And I do really want to point out it's an incredibly special lab head who will buy matching Hawaiian shirts with you in Hawaii and wear them out to a formal dinner. And I guess a really huge thank you is to my friends and family who are here today and have supported me throughout this scientific journey, even though I still don't think any of you have any idea what I do. A huge thank you to my best mates who are here today, ones who've lived with who have me come home late saying I've cleaned my lymph nodes and still don't know what that means. To the beautiful friends I've made at Wee High, it's incredible to work in a place like this and have such beautiful long-lived friendships. And sorry, I'm getting a bit fun. And again, lastly, to my lab, you've been such a great supportive team, and again, ridiculous humans, and I love working with you every day. I just want to give a quick shout out just again to my dad and Kate and James for supporting me through this journey. I know Mum would be here today if she could, and I really appreciate all of the support you've shown me. And lastly, to Phil, my partner, you're an incredible scientist, and I'm really glad that I get to come home and talk at you and you know half of what I'm saying. Thank you for deleting all the commas from my thesis, and for checking all the spelling on this today. If there's any mistakes, that's Phil's fault. But yeah, thank you for supporting me, and I really look forward to where our scientific careers go together. I love you. And thank you all to you for coming today. I've left myself a good 10 minutes for questions.
Yes, great time for questions. There are none online. Thank you, Verena, for opening your phone. Anyone else? Doug? Oh, sorry, Doug. We're home.
Sorry, I'll stop caring.
I'm going to yell it out, I guess. You had that really lovely study where you looked at your special cells near the cortical ridge, and you broadened it out to influenza and bacterial infection. So it was really interesting because at what point do you see that error on that curve to actually be at the cortical ridge? So meaning like when I looked at the influenza one, it seemed a little bit more left, and the listeria one wasn't fully at the peak of the cortical ridge, but really more to the right-hand side. So
yeah.
At what point do you make that call?
So yeah, that's a really interesting question, which has a lot behind it. And so I didn't go into it today. But what you're looking at with those three different infections is when you look at the actual makeup of the lymph node in terms of central memory versus effector, it's quite different. And so when you're talking about the influenza lymph nodes, we have most of the cells are actually quite far out into the periphery, further than the cortical ridge. They've got a higher proportion of effector memory cells in those lymph nodes when compared to listeria, which is almost all central memory. And so they're kind of just tucked at the edge of the paracortex. And it's really nice because HSV1 kind of sits between the two, and it's got these two nice peaks. And so what I think is happening is that the central memory cells are still at that edge of the paracortex, but it's the effector memories which are really the ones that are out furthest at the subcapsular sinus. And so we're kind of looking at ways to dissect this. I was trying to stain for the effector cells using a marker, CX3CR1, which we know is really expressed at the periphery in effectors, but that antibody was terrible. So one of the ways we're hoping to approach that is with the KD mice, so colouring in the memory cells that are at that very edge, and then the ones that are a little bit more closer to the paracortex, and seeing if the makeup of that's different. So yeah, that's a really, really good question. Thank you.
I just have a comment. I thought that was one of the loveliest seminars that I've heard. The images were beautiful. As director, I guess what you always hope for are there's vibrant collaboration and a real respect between people doing biology and people working in more technology-based labs like the Centre for Dynamic Imaging. It was just a beautiful seminar, so congratulations, Brigitte.
Thank you, Doug. Thanks.
Beautiful talk.
Thanks.
I was just wondering, when you skew towards your T stem-like memory cells, how does that affect infection, like the original infection? Do you see any difference in pathogen clearance or inflammation or all of the above?
That's a really interesting question. So in this, we didn't see a huge difference in viral clearance. One of the things that was interesting, though, is if we took those stem-like memory cells and transferred them into new mice, we saw that they were, I've completely forgotten my words. They live longer than cells, the other cells which are effectors. So we kind of defined that. Ben at the moment is really looking a lot more closely in his PhD at what the outcomes is of when you skew stem-like memory and how that influences viral control.
Sorry, Scott's here to ask a question.
Yeah, Scott's allocated question.
I promise it's not preloaded either.
I'm ready.
I can ask many. That's great stuff, Brigitte.
Thank you.
I'll ask a sort of a biggish question. So CXCR3 drives the cells into the cortical ridge. Then what happens? Like, what is the signal and what are the cell types that they need to see in that region to push them into the effector phase?
To push them. So are you talking about secondary challenge?
No, I'm talking about the first part of it.
Oh, the first part.
Yes.
I guess the interactions with the CDC1s out in that region, which are presenting antigen and driving that effector proliferation. I know there's been a lot of work done on that. It's been a while since I've delved back into that project. But it's really kind of that interactions with those unique subsets of dendritic cells, which are encouraging effector proliferation.
And a quick second question. So CXCR3 is really critical to drive them into that region in that primary phase. And then you see in memory, your central memory cells in the same region.
Yeah.
Do you know if CXCR3 has any role in that?
So we looked in the knockouts and it was no change. Okay. So that's why it's really interesting.
Yeah.
So yeah, this initial work came at the tail end of the work, the CXCR3 work, where we just left the mice out till day 60 and wanted to see if there was a difference, and there was no difference in location, which was super interesting and really where this work kind of stemmed from.
Yeah, great.
So my question is following on a little bit from that. So you mentioned that if you establish memory outside of the mouse and then transfer the cells in, they don't go to this area. And so I'm wondering if there's a role for really long-term antigen presentation by the dendritic cells to maintain cells in that cortical ridge. So I guess one way to do that would be to establish their location in the mouse. Sorry, my phone.
Yeah.
And then take them out and put them in a new mouse. Do they go to the paracortical ridge?
So I've done that. Yeah, so it's very preliminary because we didn't get a lot of cells from the sort. We're repeating it, but largely, it's not significant, but largely they went to the cortical ridge.
Oh, they did.
In both either a naive or a previously infected host. So it really seems like it's a cell intrinsic thing that's driving them out. So that was one of the first experiments I wanted to do, and it's just taken a while to get all the parts. It's a lot of lymph nodes to mash and sort. And
yeah, sure.
Yeah.
Okay, thank you.
No, thanks. Great question.
I can see one more over
Yeah.
here.
Oh, Ken.
Just going back to your introductory statements where different labs had totally different results, why didn't they see what you see, apart from the fact they didn't have you in their lab?
Well, I mean, the lab which had the results where they were both in the paracortex was because they had in vitro skewing. It was really interesting in the first team where they did the vaccinia virus and they showed that peripheral positioning and they threw everything they could at it. That was published pretty much just after the in vitro. And I was really interested why they didn't pursue it further because it was such a specific question. So I don't know, maybe they didn't care. But like we think it's really important. So I'm pretty excited that the research was there to do.
Anyone else? All right. Well, with that, one last thanks for Brigitte.
Thank you so much.